Okay, in the lecture I gave you a basic go over the protists. In the lab, we need to recognize them as if we were in the lab. Okay, so again, we talked about the protists and we talked about plant-like protists, animal-like protists, fungus-like protists. But you're going to have to be able to recognize them, and I'll give you a list. Okay, but there'll be basically the same pictures that we have on these lab slides, so you'll be okay. So we know we classify them according to how they move. All right, so the first one is Plasmodium, and it causes malaria. It belongs to the phylum Apicomplexa. So you do need to know the phylum name. And I suggest you make a little chart, but I'll put this on the test review. That means it doesn't have flagella, cilia, or pseudopods, but it moves by a gliding mechanism with adhesions. And the four main ones were the vivax, falciparum, malariae, valley. Again, affected bite of the female Anopheles mosquito. And we talked about the different symptoms, and that falciparum is the most serious. I gave the clue that this is bloodborne. So we have two organisms that are bloodborne. So if you're trying to figure out which is which, go look and see if it's a blood picture. It's one of two organisms. So this is Plasmodium, and you can see I mentioned in the lecture that it starts in the cell in the ring form. So this is lots of ring forms, and you can see how many red blood cells are actually infected. So if we were to do a count, we would start counting 1, 2, 3, 4, 5, 6, and count 100 red blood cells. And out of those 100 red blood cells, how many had the parasite in them? Okay, but you can see that lovely little ring form. I can tell you right now, this is Plasmodium vivax just by looking at it. It is not falciparum because it does a little differently. But the one that just forms lots of ring forms like this is Plasmodium vivax. Once they get out of the red blood cells, they form schizons, and this is what they look like. Okay, and you can see that little ring form right there. Okay, so you can still get them in the red blood cell that they begin to move out, and that's where you start to get those, the symptoms and everything. But there it is. So again, red blood cell, those little ring forms. That's what I'm going to give you on the test if I give it to you on the test. Just FYI. Toxoplasmosis. This is also an apicomplexa. Again, one of the world's most common parasites. We talked about this with the cats. Undercooked contaminated meat or exposure to infected cat feces. And again, this is what it looks like. So I'm going to pick the same pictures and say, what is this? And I'll ask you a question about it, because the lab is, when we do this, we would normally have, walk in and you had microscopes and the slides would be underneath the microscopes, and it's like, identify the organism. Okay. We can't do that, but that's what you're going to do based on these. Another apicomplexa is Cyclospora. We talked about this, the outbreaks associated with imported produce like raspberries, strawberries, cilantro, salad mix. Yeah, snow peas, basil. I got put a little more information in here for you. But Cyclospora, that's the picture. I'm not going to change these guys. So, make flashcards or whatever you need to do. And then again, another apicomplexa, Cyclosporidium. This is the one, watery diarrhea. Basically, these guys can survive for long periods outside the host and very resistant to disinfectants, notably chlorine belly stuff like bleach. So pretty resistant here. Cyclosporidium, sorry. That's what it looks like. Again, I'm going to be using the same pictures because I would have these slides. So just FYI. All right, Giardia, Lamnia. We've talked about Giardia, and just don't drink the contaminated water. Most of these cases will clear up on their own within a few weeks, but it's usually this gassy, greasy stool. Ugh. It is most common in poor sanitation, and I've already told you about the lovely, looks like lovely little kites with faces on it. But that's exactly what Giardia looks like. All right, Trypanosoma. Again, this is the other one that is bloodborne. So look at the difference. And I told you already, the trypanosoma does not go in the red blood cell. So that's the way you tell it apart. So I gave you some of the symptoms. But there's the slide. It's the exact same slide I had in the lecture. And I've just given you a little more information, a little more things, okay? Without treatment, yeah, it's generally fatal. But none inside the red cell versus inside the red blood cell. So that's how you're going to tell the two apart, okay? Trichomonas, now we're going to a different phylum. We're out of Apicomplexa, okay? And we're going into Sarcomasticophora. Aren't we having fun? These are guys that may either move by one or more flagella. That's what differentiates them. And we've already talked about this, but there is that lovely little guy, and there is that lovely little flagella. You can see them, okay? Use the same picture again. All right. Entamoeba histolytica. This is the one that causes amoebic dysentery, and you can die from this stuff. It is an amoeba-like organism. Picture here. To me, when I look for this in specimens, to me, and it looks like a fried egg. I mean, that's what it looks like. You got an organism, and you got a nucleus, and it looks more egg-like to me than it does anything else. That's may be how you recognize it. This one, don't worry about the phylum on this. This is a new phylum, so I'm not going to go crazy on this one. But I'll give you a list. This is Naegleria again. This is the brain-eating amoeba. It can get really, really nasty. I gave you a little bit more information about it. Sudden, rare, fatal brain infections. Can kill you in just a couple of days. And again, the same picture that I gave you before. And you can see the lovely little white cells. Those are little white cells. And there is the Naegleria. Okay. And that's it. So you have these slides, and I'll give you look. I would suggest studying for this, making flashcards. Print the picture, put the picture on the front, flip it on the back, and just put the name of it and a little bit of information, and study for that. And that's the easiest way to study for these guys. Okay.